Machine learning is quickly becoming an absolutely essential tool for modern biology. So, in this explainer, we're going to take a look at how it actually lets scientists make sense of the incredible complexity of biological data.
You know, the scale of data in biology today is just mind-boggling. To put it in perspective, a single human genome contains over 3 billion base pairs. And that's just one example of how these data sets have exploded in size and complexity in recent years.
And this, right here, is the central challenge. When you have data sets this massive, how can a researcher possibly dig through all that noise to find the actual, meaningful biological signals? Well, this is exactly the problem that machine learning is built to solve.
To really get a grip on how machines learn, it's actually helpful to start with something we're all familiar with: how we learn.
The source material gives us this perfect analogy. Think about a child learning to catch a ball. They don't need to understand the laws of physics, right? They just learn through trial and error. Through observation, they build this internal model that gets a little bit better with every single throw. That's the core idea behind machine learning, building a predictive model by testing it against data and constantly making corrections. So, the formal definition of machine learning is really just that. It's the process of fitting these predictive models to data. It's a way to computationally recognize patterns, and it's especially critical when the data is just way too big for any single person to wrap their head around. Okay, so within the world of machine learning, the approaches are generally split into two main toolkits, and which one you use really depends on the kind of data you're working with. So the first big distinction is between supervised and unsupervised learning. In supervised learning, you're working with data that already has labels, a known ground truth. So for example, you could train a model on thousands of proteins where you already know the final structure. But with unsupervised learning, you're working with unlabeled data. The goal there is to find hidden patterns or clusters on your own, like grouping patients together based on similar gene expression, without knowing their disease status beforehand.
The second key difference is between what we call traditional machine learning and deep learning. Traditional methods, think support vector machines or random forest, tend to be faster, they're often easier to interpret, and they can work really well on smaller, well-structured datasets. Deep learning, on the other hand, uses these very complex neural networks. It really shines when you have just massive amounts of data with really intricate patterns, like you see in genomics or imaging. The trade-off, though, is that it can sometimes be a bit of a black box, making it harder to understand exactly how it's making its decisions.
And that brings us to a really crucial part of the whole process. You've got to choose the right tool for your specific kind of biological data. You know, the nature of your data is what really tells you which model to use. Is it an image, is it a sequence of letters, or is it a network of connections? Each of these data types has a model architecture that's basically tailor-made for it. Let's start with data that has a spatial grid-like structure. For this, we use something called convolutional neural networks, or CNNs. These are the absolute gold standard for image analysis. And in biology, that applies to a ton of things, from classifying different cells in microscopy images to finding patterns in DNA-protein interaction data. Okay, next up, what about data that comes in an ordered sequence? Well, for that, recurrent neural networks, or RNNs, are the tool of choice. Their whole design is built to process information where the order really matters. This makes them perfect for analyzing things like gene or protein sequences, or for tracking how gene expression changes over a period of time. And finally, for data where it's all about the connections and the relationships between different entities, we use what are called graph convolutional networks, or GCNs. This is the ideal way to model things like protein-protein interaction networks, or even the structure of a molecule, where the whole network of atoms and bonds is what defines its properties. Now, just having the right tool is not enough. You have to apply it correctly. This is absolutely critical for getting results that are valid and that other people can reproduce. A really rigorous workflow is non-negotiable. It all starts with preparing your data, and then splitting it into 3 distinct sets: a training set to build the model, a validation set to fine-tune its settings, and a final test set. And the absolute most important rule here is that you use the test set only once, right at the very end. This is the only way to get an unbiased look at how your model will actually perform on brand new data it's never seen before. If you don't separate your data correctly, you can run into a massive and very common problem in biology called data leakage. So what is data leakage? Well, it happens when your model isn't actually learning to generalize. It's just memorizing the training data.
A classic example in biology is if you have two very similar, evolutionarily related proteins, one in your training set and the other in your test set. The model will look like it's performing brilliantly on that test protein, but it's not because it learned a general principle. It's just because it saw something almost identical during training. And this, of course, leads to you thinking your model is much better than it actually is.
All right, so let's shift gears and look ahead. Beyond just analyzing the data we already have, the next big frontier for machine learning in biology is all about creation. The entire field is starting to move from predictive models to generative ones. So instead of just predicting a protein's function from its sequence, scientists are now starting to use models to generate completely new protein sequences that are designed from the ground up to have specific functions. One of the key tools for this is something called an autoencoder. What it does is it first learns to compress data, like a protein sequence, into a much smaller, compact form, and then it learns to decompress it back to the original. Once it's trained, you can use that decoder part all by itself to generate brand new, synthetic proteins that have literally never existed in nature.
So just as a quick recap, this chart really sums up the main theme here. It's all about matching the right tool to the right task. Whether you're using a traditional method like a random forest because you need to understand which features are most important, or a deep learning model like a CNN for image data, the choice is always driven by your data and the biological question you're trying to answer.
And that leaves us with one final, pretty profound thought. As these generative models get more and more powerful, we're really moving beyond simply reading the book of life. The question is no longer just about what we can predict, it's about what we can create.